So good day, everyone. So this is Module 6, Carbohydrates. I am Dr. Erwin Abrencilio. So right now we are going to talk about these learning outcomes. First, determine the general properties of carbohydrates. Second, explain the functions of carbohydrates. Third, differentiate the three major classes of carbohydrates. Fourth, describe carbohydrates based on their structures and formula. Five, explain the concepts of chirality, enantiomers, stereoisomers, and DNL families. And last, recognize the biological importance of carbohydrates. So these are the target learning outcomes that you should accomplish at the end of the module. So this module is intended for 2 weeks. And as I have mentioned in our CNID, that the deadlines of submission is during the class schedule and the submission is through a hard copy. Most of the answers on the activity sheets are formula writing as well as naming, no? So you need to really have a hard copy for that. So let us talk first, lesson 1, general properties of carbohydrates. So what are carbohydrates? So first, carbohydrates act as energy reserve. So meaning to say that our body, as we need energy, we always get carbohydrates or the sugar. So that is also the reason why we can easily access sugar in our body. For example, the glycogen that are stored in the liver, no, are converted into glucose whenever there is a low amount of glucose in the blood. And of course, it also store as fuel. We can easily access or we can easily use carbohydrates as a quick source of energy. For example, you are feeling confused or you are feeling dizzy because of lack of sugar in the blood. So you just simply eat chocolate or candies so that you can be able to access quick source of energy. And carbohydrate also act as metabolic intermediate. So as we go along with the discussion, you can be able to see that there are a lot of metabolic intermediates that are connected to carbohydrates. So for example, ribose and deoxyribose sugars form a structural frame of the genetic material, the RNA and the DNA. So aside from those sugar that we tackled in one of our modules, which is biomolecules, no? Wherein we tackle about fructose, galactose, glucose, and so on. So another two sugar that are very important in our body are ribose and deoxyribose sugar. So these are found in the genetic material, RNA and DNA. So the ribose sugar is found in RNA and the deoxyribose sugar is found in the DNA. Additionally, polysaccharides like that of cellulose are the structural elements in the cell walls and bacteria in plants. So polysaccharide are sugar, no, or carbohydrates. A very good example of that is cellulose. Cellulose is the one found in the cell wall of the bacteria in plants. So it is very rigid. It gives the shape, no, it gives the cell its shape. So of course we are going to tackle a lot about monosaccharide, disaccharide, and polysaccharide as we go along with this module. So carbohydrates are linked to proteins and lipids that play important roles in cell interactions. So meaning that carbohydrates are essential so that there will be for the cell interaction, which of course are linked with that of other biomolecules like that of proteins and lipids. And carbohydrates are organic compounds. Of course, it is composed of carbon. And since they are organic compounds, they are classified as aldehydes or ketones with many hydroxyl groups. When we speak of hydroxyl group, these are those things that you can see here, the OH group, OH, like this, no? And of course, for you to be able to tell whether it is an aldehyde or a ketone, sugar, all you have to do is to look on the functional group. So on this side, let's see, I'll be using highlighter. So this one, this is CHO, so this is an aldehyde. And this one, C double bond O, this is a ketone. So meaning that um this one, the first example is a glyceraldehyde. So that's the IUPAC name, if you can still recall how to name aldehyde. No, so this is glyceraldehyde, which is considered to be as an aldose. So the aldose term is the name as a sugar. So OSE, if you'll notice that when we speak of sugar, the end suffix is OSE. And then it is an aldehyde, so there is the prefix ALD or ald plus the OSE as suffix, so forming the word aldose. On the other hand, the dihydroxyacetone, same number of carbon with glyceraldehyde. So there is a ketone here. So the ketone can be found in any part of the structure except on the both sides. So because if you are going to put ketone on the either of the both side, it will become an aldehyde. So ketone exists between the carbons of the opening and ending carbon of the compound. So this is a keto. So from the word ketone, you'll have here KET, and then OSE for sugar. So the name is keto. So this is an aldose, and that one is a ketose. Okay, so let us now talk about lesson 2, chirality of molecules. So I am sure that you are still familiar with this topic since in our previous module we talked already about chirality or the chiral center. So if you will recall, for us to tell whether a certain atom is a chiral center, there should be 4 different atoms that are bonded to it. So since we are talking about the carbon, so carbon is a chiral center if there are 4 different elements that are bonded to it. So most monosaccharides exist in 2 forms, left-handed and right-handed form. For the L isomer for left and D isomer for right. So you just think of your hand. So you have your left hand and the right hand. So monosaccharide exists the same way. So you have there the left-handed monosaccharide and the right-handed monosaccharide. So chiral center in monosaccharide would mean 4 different atoms of elements are bonded to the center carbon atom. So later on, I'll be showing you the structure of the monosaccharide with the chiral center. So there are 2 types of objects. Generally, no, I am not referring to monosaccharides only, but in any other objects that we have. So there are what we call the superimposable on their mirror images, or known as a chiral, and the non-superimposable images or chiral. So just like this one, this could be a mirror image, no? So the one on the left is and its mirror image is the one on the right. So with this, you can be able to see that the carbon is a chiral center because there are 4 banded atoms, different atoms banded to the carbon. So we have here the OH group, the CHO group, the H, and then the CH2OH group. And the same manner on the other side, on the right side, which is the right side is the mirror image of the left side. If you'll notice, the left is OH, the right is OH. The here is the right is H and then the left is H. And if you are going to look into the Fischer projection, this one at the bottom is known as a Fischer projection. So you have here OH on the left and H on the right. So it's the opposite of the one on the right, OH on the right and H on the left. So this one, this OH, no, is very, it is very important in analyzing the isomerism of the monosaccharide. So later we'll talk about it more. So classification of the molecule based on the rotation of the plane polarized light. Though I did not mention anything about this one, how to determine or what is polarized light. So I will just go directly to the point that monosaccharide sugar are either D-isomer or L-isomer. So pag sinabi nating D-isomer, ang ibig sabihin ng D is dextrorotatory or simply D means right. And L, levorotatory or L-isomer. So you just think that L is left so that you will not be confused. Again, L is left and D is right. So with that, you can be able to see here, no, that we have here an L-isomer on the one on the left and D-isomer, the one on the right. So how do we determine that? So as I mentioned, this is L and this is D. So L and this is D. So what's the basis of telling whether the molecule or the monosaccharide sugar is an L-isomer or a D-isomer? So in this case, ang tinitingnan or the one that we can observe is this OH. So the OH that is next to the bottom carbon, this one. Okay? So this one next to the bottom carbon. Okay? So that one. So since this OH is found on the left side of the structure, so we call it as L-isomer. Okay? Or left. And then this is found on the right, so we call it as D-isomer. So no matter how complicated the structure is, all you have to do is to look into the OH near the bottom carbon and determine whether it is found on the left or found on the right. So if it is, again, if it is found on the left, it is L-isomer. And then if it is found on the right, it is OH isomer. Well, of course, aside from that, you have to name it. But later on, we are going to discuss how to name the specific monosaccharide or whatever the sugar is. Okay, so let's talk about stereoisomers. So way back in your organic chem, I think this is one of those one of the topics there. So what are they? So they are isomers that have the same molecular and structural formulas but differ in the orientation of atoms in space. So if you are going to look back about isomers are they have the same formula but of different what they call that, different structure, no? So in stereoisomerism, it has the same concept. So there are two types, enantiomers and diastereomers. So enantiomers and diastereomers are quite different, no? But they are both stereoisomer. So what's the difference between the two? So enantiomers are stereoisomers whose molecules are non-superimposable mirror images of each other. So they are known to be as molecules with chiral center. On the other hand, diastereomers are stereoisomers whose molecules are not mirror images of each other. So You have to remember, enantiomer, mirror image, diastereomers are not mirror image. So, just imagine yourself in front of the mirror. When you raise your right hand, the one on the picture or the one on your reflection will raise its left hand. And when you raise your left hand, the one on the mirror will raise its right hand. So if the molecule is the mirror image of the the first molecule, so we call it as enantiomer. However, if it is not a mirror image of it, we call it as diastereomers. So they have more than one chiral center, but it doesn't mean that it is not a an enantiomer. There's no chiral center. There is still chiral center because what we are talking here is the stereoisomerism. So they are both stereoisomers, but they are not mirror image of each other. So to sum it up, the stereomers or diastereoisomers are stereoisomers that are not enantiomers. They are non-superimposable imposable mirror image of each other. So diastereomers can have different physical properties and different reactivity. So in another definition, diastereomers are pairs of isomers that have opposite configurations at one or more of the chiral center but are not mirror images of each other. So let us look on the next slide so that you can be able to look into the example so that all these explanations can be applied to that. So we have here three samples. So this is tartaric acid. This one is the one here on the left is an L-tartaric acid. So this is L-tartaric acid, and this D here on the right is a D-tartaric acid, and this one on the right is the mesotartaric acid. Don't mind the name, no? We are not here to learn the name of these three specific acids, but this will give you a glimpse, or this will give you an idea what enantiomers and diastereomers are. So this is our original molecule, no? So this is tartaric acid, and this is in L, no, in L form or left because of this one. This is left. Okay. Now, the one on the right is an enantiomer of the L. Enantiomer because it is a mirror image of it. Look at this. So this is H on the left. This is H on the right. And this OH is found here on the right. And on the other hand, it is found at the left here. And the OH here is found at the left. And this is found at the right. And this is H found at the right. And this is found at the left. So in that case, it is the exact reflection of the L-tartaric acid. So therefore, you can see that an L form or L isomer, its enantiomer is the D form. If the given is in D form, its opposite is in L form. Okay? In this particular example. Okay? So where is now the stereomers? So the stereomers here is found here at the right. So this is the original, the one on the left, and this one, mesotartaric acid, is not a mirror image of the original. Why? This is found. This H is found at the left, and this H is still found at the left. So in this case, it is not a mirror image of it. And this OH is found here at the right, and this OH is still found at the right. So in this case, it is not a mirror image. And this is found at the left, the OH. This is found at the right. Okay. So this one is okay. This part H at the left, and this is H at the right, and this is H at the left. Now, though this part, no, the last part are mirror image, but the entire molecule, no, because of this structure here at the bottom of the COH or the second carbon, no, is not a mirror image of the original. So then, therefore, it is known to be as diastereomer. Okay? Now, as I mentioned a while ago, um, enantiomer has one chiral center. Of course, that depends on, no, on the structure. And then we also have, meanwhile, if it is diastereomer and there are more than one, no, so there are more than one chiral center. So in this case, let us look into this one. So this is chiral center, no, this one. So we have here the COH, and then the C here, and then the OH here, and then the H here. Okay. On this side, we have here two. Okay, so we have one, and then two, another carbon here. Okay, so meaning to say that whatever the stereoisomerisms are, still there will be chiral center because we are talking now about the monosaccharide sugar. All right. So let us now look into this specifically. So we have here enantiomers, diastereomers, and epimers. So let us include the epimers so that later on you can easily understand what they are. No. By the way, you'll notice here that there are asterisk. Sorry, asterisk here. So they are called as um chiral center. Okay. Oh, chiral center here. So there are a lot of chiral centers. So again, enantiomers are mirror images. So for example, you have here the L-glucose and then the D-glucose. So this is they are enantiomers of each other. Okay, so glucose here. So this is the glucose on this side. So it is a complete mirror image. This one naman is diastereomers. This is D-mannose and D-galactose. Um, Again, for those who are still confused on how to determine the L and the D, so this is L. So this is OH bound at the left. D is OH bound at the right. D-mannose, this is bound at the right. D-galactose, OH is bound at the right. Okay? Now, they are stereomers because they are not mirror image of each other. So let's see. This OH at the left, OH at the right, OH at the left, OH at the left. So this one, you can see here that they are both at the left, and as well as this one. So because of these two structure in this formula, no, of D-mannose and D-galactose. So they are diastereomers because they are not mirror image of each other. Now, let us look into glucose or the epimers. Okay. So what are they? So epimers, two sugars that differ in configuration at only one chiral center. So D-glucose and D-mannose. So D-glucose and D-mannose. So D-mannose has an epimer of in first carbon. So this in the second carbon. Okay, so this is the original. Look at that. So H is found at the left, OH is found at the right. Okay, so going back, D-mannose is an epimer, has an epimer in the carbon 2 from the glucose. So why? Because this one they differ here. This H is found at the left, and this is found at the right. And OH here is found at the left, and this is found at the right. But the rest are the same. Okay? They are the same. So therefore, D-glucose and D-mannose is an epimer, has an epimer from D-glucose on the second carbon. So we call it a C2 epimer. Okay? So C2 epimer because it is found on the, it's difficult to write here. Okay. So I hope that you are now having an idea what how to analyze enantiomers, the stereoisomers, and epimers. Okay, so more about epimers. So this one shows the glucose structure here at the center, no? So this is glucose structure, and then you have here D-mannose and D-galactose. So D-mannose and D-galactose are both epimer of glucose, wherein here, no, it is highlighted already. So epimer at C2, and then this one, galactose is epimer at C4 because that is found at the fourth carbon. Okay, so that's how you analyze epimers. Okay, so as I mentioned a while ago, monosaccharide sugars can be expressed by a Fischer projection formula. So if you will going to recall in your organic chem, so there are a lot of formula like the molecular formula, and then the condensed structural formula, the line angle or the skeletal formula, and so on. Now, what is a Fischer projection formula? So it is a method for giving molecular chirality specifications in 2 dimensions. A Fischer projection formula is a 2-dimensional structural notation for showing the spatial arrangement of groups about chiral centers in molecules. So you can easily observe the chiral center when the structure is in a Fischer projection formula. So the 4 groups attached to the atom of the chiral center assume a tetrahedral geometry, and it is governed by the following conventions. So like this one, okay? So this is the Fischer projection formula at the right, at the left. This one, this is a Fischer projection. And this one is the usual formula. Like and then this one is the ball and stick model of the formula in the Fischer projection. Yeah. Okay. So how to convert? No? So you have here convert the Fischer this formula in a Fischer projection. So simply you just draw a line, a cross line for this example, and then just copy the location of the C of the OH and the CHO and CH2OH. So you have here the one here, it is CHO, it is found at the there on top. Then H here at the left side, and then H here at the left side, and then OH here at the right side, then OH here at the right side, and then the CH2H, you just have to copy it. And then here. So of course, it will follow the same formula. This is D glyceraldehyde. So the Fischer projection is also in a D glyceraldehyde. Okay? So that's the end of the lesson 2. So this time, let us talk about the classification and functions of carbohydrates.